In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the JAX Laboratory.
Hi, and welcome to the first JAX Tech Talk, or commonly known as JTT of 2023. My name is Hannah, and one screen over is my colleague Neeraj. We both work as technical information scientists at the Jackson Laboratory. For those of you who are seasoned viewers of JTT, hi, it's good to see you all again. And for those of you who are new, welcome to this fun 15-minute informal chat session where we get together to discuss all aspects of science, ranging from troubleshooting that pesky genotyping assay to new cutting-edge research and models. I emphasize we because you're part of the discussion too. Please submit your questions in the chat Q&A function throughout the talk. We will answer as many questions as possible at the end of the presentation.
Hello, everyone. My name is Neeraj. Let's get started with the topic for today's discussion. As you all may know, February 28th is Rare Disease Day. So in honor of that, today's JDT is all about the past, present, and future directions of rare disease research and how JAX is here to support. A rare disorder is a disease or condition that affects fewer than 200,000 Americans. Altogether, there are more than 7,000 rare diseases affecting more than 30 million Americans. Many rare disease conditions are life-threatening, and most do not have treatments. We are very excited to be joined by Dr. Kat Lutz. Dr. Lutz is a leading expert in the development, characterization, and therapeutic testing of rare disease mouse models, and is the Vice President of the Rare Disease Translation Center here at JAX.
So thank you, Kat, for joining us today, and we're very excited to be able to pick your brain about all things rare disease.
Happy to be here.
So let's go ahead and get started with some questions.
Sure.
We mentioned in the introduction that you are the Vice President of the Rare Disease Translational Center at JAX. I view the Translational Center as really this melting pot, bringing together the scientific community and the individual patient foundations. Could you elaborate further about why the Rare Disease Translational Center has brought these two communities together, and also share a little bit about the overall focus and mission of the center itself?
Sure, I'd be happy to. More and more, we are working with patients and patient foundations where rare diseases and their diagnosis is recently come to light. It's often the case that patients are on this diagnostic odyssey for a very long time before they get a diagnosis, and when they do, they are now faced with another hurdle to conquer. And that is, what are they going to do with this knowledge, and how do they tackle the research, and how do they tackle the therapeutic components of it? So we've definitely seen patients and patient foundation organization extremely educated in this area, and understanding that their quickest path to translation is really to get the research and to get the preclinical discovery phase started and initiated. And that's really where we come in. We facilitate those discussions. We're sort of guides, if you will, on what the next steps are for individuals. And we try to work with them to see if we can't get their research kickstarted through not only the work that we do, but the collaborators we have across the biomedical research community.
Thank you for explaining us the focus and mission of the Rare Disease Translation Center. Kat, what would you say about rare disease patients and organizations that is different now than it was, say, 5 years ago?
Yeah, I think that's a great question, and I think one of the reasons why we're really excited for the rare disease community, especially at this given time, is that I do think that diagnostic odyssey is changing a little bit. It's definitely shortening. Information and access to diagnostic sequencing, whole genome sequencing, is putting a diagnosis in patients' hands much sooner than it had in the past. And then with that, on the research side, there have been so many advances in new technologies that we're currently, looking squarely at and understanding exactly how forms of, gene therapy, genetic medicine, base editing, antisense oligonucleotides can help not just patients who have, I'm going to say common rare disease, but even individuals, where there's an N of 1. I think that there's a lot of promise, in those kinds of components. And from a research side, we have never been better poised to deal with this than we are today.
Great. Thank you for explaining that part, that there are now resources available for these families, and that's playing a very important role in helping the rare disease patients. My next question is, and I have heard many times from you that that after the diagnosis, everyone needs a mouse.
Yeah.
So can you explain why that it is?
Yeah, absolutely. And it's one of the time points in the patient's diagnostic journeys, and when they do reach out to us, when they do have that diagnosis. And I think really what they're starting to understand is that, whether you understand the mutation, the mechanism of action, if you need more research that's done, or if you just need that patient avatar, if you will. Sometimes in rare diseases, we just don't have enough patients within a given disease area, or the heterogeneity is just so great that it takes, it's not as easy to figure out what exactly is happening with those patients and those mutations. So by building a mouse model, an in vivo mammalian system, we can mimic those patient mutations in the mouse models, better understand things like the mechanism of the disease. But I think also it gives us that, as I said, that avatar for clinical, preclinical testing of therapeutics, so that if we do have some hypotheses about an antisense or gene therapy, we can start to answer questions using those mouse models about, exactly what is needed to correct or relieve some of the symptoms of that disease or correct that disease. And so that's really where the mouse model comes in. It sort of substitutes, for the patient population in that regard.
Yeah, that makes sense. Kind of piggybacking off of Neeraj's question, since everyone needs a mouse model, then how does JAX kind of work to solve the mouse resource issue for the rare disease community?
Yeah, that's a really great question. We do have a lot of mouse models, and we do have a lot of resources. And I think most importantly, we've had some really great advances in technologies that allow us to make mouse models and genetically engineer them much faster and much less expensively than we did it before. So going from, wanting to have a patient mutation engineered in a mouse model is not as big of an obstacle as it was before. So that's really helpful. And in addition to that, we have this incredible mouse repository here at Jackson Laboratory, where we just have so many existing mouse models that we can import and use for that. But I do think, it's the genetic engineering technologies and our resources and able to, utilize those genetic engineering, as well as a lot of NIH funded grants, that help us to facilitate that. So the funding is a big part of it. So you put the funding and the technology together, and that part's much easier than it has been in the past.
Yeah, that makes sense. So we have one last question for you before we turn to the audience to see the live Q&A. To you, what is the most exciting development on the horizon for rare diseases?
Oh, I think it definitely has to be in the genetic medicines that are coming on the horizon. It's just amazing to think of technologies like base editing, which almost sounds, science fiction, that you could actually go into somebody's DNA and fix a mutation that is there. It does, right? It sounds like a comic book, science fiction novel, that maybe 15 years ago someone dreamt up. But those are really starting to become realities. And I think not all the technologies are there. They're not all put into practice and in place, but we've had some pretty big wins, in the past, SMA being one, and other rare diseases where gene therapy and antisense oligonucleotides have really, started sort of opening up the door for what's possible. And then I think the technologies that, help perfect those, with better deliveries, capsid designs, and more, component strategies will just get us there a lot faster than we have been before. So I'm really excited, to see where these technologies are going to lead and how far we can advance them, for the next couple of years.
Yeah, that sounds really, really exciting. Neeraj, do we have? Oh, go ahead.
I think I will go on with adding one more question to that. So we have talked about like what are new things we have available for the patients, like new research tools and techniques. So what is going to be the biggest challenges still ahead in rare disease patients? And what are some of the actions the community is going to take to solve them?
Yeah, I think that there are hurdles and challenges still ahead. I think that the discovery phase, the preclinical work that I've just described about mouse models and cell-based models and the research, in some ways, it's not the easy part, but in some ways it is maybe a little bit more refined in terms of the pipelines. I think some of the hurdles that we still have are costs in manufacturing, costs in toxicology. It's a very expensive proposition to get, a therapeutic into the clinic. And when you have, in some cases, very small patient populations, it becomes a challenge, I think, to think of like, what is that business model? It's certainly not going to be treating thousands of patients because there aren't thousands of patients. So I think we need as a community, to sort of come together and try to figure out, how we do that a little bit better. And I think it really is the patient advocates, who are going to be the main people, who steer the ship and get us moving in this direction, because we need, really crowdsourcing at every level, whether it's, the hospitals that do the clinical trials, the FDA that helps us to really understand what they're looking for from a regulatory standpoint, and then more efficient, effective ways to do manufacturing and toxicology. It all needs to come together. And I think it is getting there, but those are hurdles that are still, in front of us that we all collectively need to work on, for sure.
Kind of going along with that question as far as, costs and everything. And this is actually something you mentioned in one of the earlier questions of that there's common rare diseases. So what do you mean by a common rare disease? Is there a range of rare diseases in this? Actually, someone from the audience asked, what would you consider a common rare disease versus a true rare disease?
Yeah, I think this may be a little bit of internal jargon, but it really refers to the number of people at any one point in time that are affected with that particular mutation. So, for example, Duchenne muscular dystrophy is a disease that, as devastating as it is, it affects a large number of people. I think Friedreich's ataxia and some of the more common ataxias are still considered rare given their genetic mutation, but collectively they have just a little bit bigger of a patient population than, for example, a de novo mutation that might occur in an individual where they literally are the only, or figuratively, really the only person in the world that we know of that has that mutation. So those are what we call our N of ones, or our nano rare diseases, but they really do affect a very, very small percentage of individuals, and they're a little bit harder to tackle because collectively, if the mutation like Duchenne or myotonic dystrophies or some of those have been around, they're inherited, they're well studied, we know a lot more about them. Than we do some of these nano rare diseases and these really ultra rare diseases.
So I think we have one more question in our Q&A. So how do you decide if making a new mouse model is appropriate for each given disease?
Yeah, that's a really good question, because we do get approached a lot by the patient communities, and we certainly understand their need and their plight, to have that patient avatar. A lot of it is just a little bit of research on our part. We really need to understand the disease, and we need to understand, what the mutation is. A lot of times the mutations are causative and known to be causative, but sometimes they're variants of really unknown significance. And then we kind of want to do a little bit of extra sleuthing to make sure that we understand that mutation really is causative in that regard. But then we really sort of look at what we currently have for mouse models. Again, we have these large numbers of repositories, and sometimes the answer already is there. If it's a loss of function mutation, or even a missense mutation that occurs, we can kind of glean a lot about what it is we need there. And when we always are thinking about sort of the therapy and what is known about the research in that space, to begin with. Thinking about the therapeutic strategy in mind, for example, a splicing mutation that you might be able to address with an ASO might be a little bit easier to tackle than a more complex genetic mutation that might not leave us with that many options. And so it's not the only thing that we think about, but I also think we are considering the unmet need in that patient population as well. There are some diseases where there are multiple mouse models, and then there are places where there are none at all. And so we like to try to get the conversation started, especially for those rare diseases where no mouse models exist or just aren't representing the patient population very well. The idea is that if you can get the resources like the mouse models out there, it sort of lowers the bar or the barrier, for other people to sort of jump in and start working on, that disease and working with those mouse models, because all the mouse models that we make are just readily available to the scientific community. And that's really our plan and our strategy. We can't necessarily work on everything. But if we can provide the resources, for people first and foremost to think about, how other people around the world could take those mouse models and learn at the same time, then that just amplifies, what we're doing for those patients.
Yeah, that's really cool. If I can ask one more question, I feel like we have a few minutes left. Once a mouse model is made, then how do you determine if that model is clinically relevant for that disease?
Yeah, that's a really good question, because sometimes it's not. And we often, use the phrase, mice are mice and people are people and people aren't mice and mice aren't people. And it sounds like it's a Dr. Seuss, but at the end of the day, sometimes what we like to see are the clinical presentations and the patients. We like to see those reflected in the mouse models that we make. So whether they're seizures or muscle weakness, a form of neurodegeneration, like I said, hypotonia. There's lots of things that patients present with, especially intellectual disabilities and delays. That's often how a lot of times, especially in our younger patients, they start to understand that there's something wrong when developmental milestones are just not being reached. And so that's when patients go out, they're looking for their doctors and their pediatricians. They come up with a diagnosis, and then they often come to us. And it's not always the case that we make a mouse model that 100% absolutely reflects the patient disease. Sometimes it doesn't at all. And then we have to, when it does, that's great. We have something to work with right away. And then we're, off to the races, so to speak. When it doesn't, we have to start thinking about why it doesn't, what might be a little bit different between the mouse and the patient. And then sometimes it's just a matter of looking at genetic backgrounds and making some refinements to the mouse models. It doesn't always work, but it's not the end of the line when we don't necessarily have a patient. And then, of course, I'm neglecting to talk about, cell-based models as well. Sometimes in patient-derived iPSCs or the iPSCs that we can engineer here, we'll often find a cellular phenotype that we don't necessarily see manifesting outwardly through behaviors or other functions in the mouse.
Yeah, there's a lot that goes into deciding what it sounds like what model, and then deciding if that model is clinically relevant. Well, that's all that we have time for this week. So thank you again, Kat. This conversation has been really informative.
You're welcome.
And-
Thanks for having me.
And thank you everyone for joining us. If you have additional questions for us, or your question was not answered, please send your questions to mystack@jax.org for assistance. Our next JAX Tech Talk is on food for thought, topics on research mouse diets. Will be live on March 14, 2023. Follow JAX Tech Talk on LinkedIn so you are notified whenever we have a new episode, or subscribe to our playlist on YouTube and watch all our previous JDs. So this is Kat, Lutz, Hannah, and Neeraj signing off, and wishing that you all stay healthy, stay safe, and stay excited about your research.
Thank you.